like to highlight the works which we are currently in. So we are mainly, our primary research area of research is host microbiome interaction in pancreatic diseases, where we are actually, my special interest is gut microbial crosstalk with the host via metabolites. And our long-term goal is development of therapeutic as well as biomarkers using gut microbial metabolites and microbial manipulation by FMT and diet. So he is Dr. Talukdar, who is the lab head of us, and these are my lovely teammates. So moving on to our research, like how do we get into the microbiome research? We have, in 2012, we got a project from Department of Biotechnology, Government of India, to study the microbial diversity of tribes of India. So we realized that although like after having human microbiome project, we have a lot of data from the west sides of the world, but in India, we do not have that data yet. So we started with our microbiome profiling of the tribes. So we recruited those tribal population who still possess the ethnic food habits, and we found out that microbiome profile of these tribes are very much unique than the western part of the world. In 2016, we have again another set of Indian scientists who have reported the similar results like us. They have reported that our Indian gut microbiome is unique, as well as the microbiome of our tribes are similar to the Southeast Asian tribe. Then we also cross-check how Indian gut microbiota look like. Here we have like taken into consideration all the studies from the different parts of India. And the take-home message of this study, we found that Prevotella copri is most abundant in bacteria in Indian gut, which we did not see in Western population. And also microbiome, like we found that again, Indian tribal gut microbiome is similar to Mongolian gut microbiome. So now moving towards the gut pancreas axis, since we now know that microbiome profile of Indian gut of healthy population of India. So now we want to know how this microbiome profile is, microbiome is having the association with the pancreatic diseases. So we have this beautiful paper in Nature which says that healthy pancreas do have bacteria, and then coming up to all this disease association with microbiome. So generally, microbiome associate with host via metabolites. So we want to know how the metabolite profile looks like along with the microbiome profile of the host. So I'm going to like briefly elaborate how pancreatitis look like. This is a pancreas, and these white dots or white things are stone or calcium deposition. Generally, the disease having clinical setup have a three main scenario. One is chronic pain, another is malnutrition, and 50% of this patient having pancreatitis are having diabetes. So we thought that since it is a diabetes and only 50% of chronic pancreatitis patient developed like diabetes, so we want to know that apart from their genetic makeup, what is their microbiome profile looks like. We studied their genetic makeup. We did not find any significant differences, but we do find the significant differences in microbiome aspect. So this is the study reported by our lab. And along, like after that, we have 3 or 4 more studies which has been reported saying the same story, that we have altered Firmicutes and Bacteroidetes scenario, altered bacterial diversity, and reduced good bacteria in disease patients. So this is the paper which we want to highlight by showing the reduction of bacterial diversity and also reduction of Firmicutes in the disease cohort. Well, now the story behind, we want to know that we knew that the bacteria are different, or bacterial diversity has been lost. Now, what are the functions they are doing? If those bacterial profile are different, then in which way they are affecting the disease patients? So we found out, this is an in silico approach. We found out that these are the pathways starting from peptidoglycan, lipopolysaccharide biosynthesis are upregulated in the disease cohort. So this is the study. Since it is in silico study, we did not include any metabolite parts over like in that study. So then we moving on to the metabolite aspect. So then we consider their blood metabolite profile. We started with the untargeted approach, and we found that mostly amino acids and branch chain fatty acids are mostly altered in the disease group. And talking about the pathway, these are the 20 different pathways which has been found upregulated or significantly altered in the disease group. So we then started recruiting more patients to know whether the same notions are exist or not. So here we are using target-based plasma metabolites by using a kit from a renowned company. So when we plotted multivariate plot using the plasma metabolome which we observed in the patients and also healthy volunteers, so we found that if you see, these balls are like differently clustered, and the color coding for the different groups. CP indicates the disease group and green for the healthy controls. So we then also subgrouped all these patients based on their etiology, like whether they're alcoholic or smokers or idiopathic. So we found there are some significant differences also exist in terms of plasma metabolome if we subgroup them based on their etiology. So moving on to, after having those metabolite information, so what we started to having our main goal, which is to know the interactions between microbiome and metabolome. So we used Spearman correlation to check the association. Then we visualized in a network plot. So what we found that, just a second. So we observed positive correlation of alanine with these two particular bacteria, that is Bacteroides and Luminococcus cereus. Alanine is also known to improve insulin secretion. So what happened for in diabetic group, although like in diabetic CP group, they have reduced alanine as well as reduced bacteria, like both of these are also reduced. Again, the positive correlation we have observed with myoinositol and Bacteroides and Ruminococcus bromine. So these two bacteria have like generally found to be have a good, very much a positive role in insulin secretion. So we are thinking that the less myoinositol, like since this bacteria is less, so they are also affecting the metabolite level, plasma metabolite level, which we can later on use as a biomarker to predict that which set of chronic pancreatitis patients are going to develop diabetes in near future. Although we need more data to have any conclusion for that. So since I already explained this is a small talk, we are very basic into metabolite field and we are trying to spread our wings in that. So a summary, we have like all our research work have shown that we have altered metabolites along with the microbial dysbiosis in the disease group. And also we are looking for, with the razors, we are like hoping that we can actually create a biomarker panel based on plasma metabolome and metabolite tracing or tracking if possible to predict the disease severity as well as designing the potential therapeutics in the diseases. So I'm going to complete my talk with by acknowledging our funding agency, Asian Healthcare Foundation, Wellcome Trust, DBT, and our host and our foundation, Asian Healthcare Foundation, as well as Asian Institute of Gastroenterology, which have provided us the space to do our research. And also, thanks a lot to you, so that like for giving us the opportunity to showcase our work. Thank you. I'm open for any questions.